All right, good afternoon. We've got a few things in class to finish up about hemoglobin. First of all, we'll talk about the graph. Second of all, we will talk about medical applications. And so I want to take the time to be able to tell you the details about this, and that's why I am doing it from my office. So let me first call up the talking about the graph. Notice how we talked about in class, hemoglobin has a different curve from myoglobin. It's sigmoidal, not and it's more complicated than a hyperbola. For a hyperbola, I could just give you the calculation because it's it's got 4 terms in the equation. Sigmoidal will require a little more calculation, but we actually have one way to deal with it, but it's not quite the same way. We'll show you what that is in just a second. But the first thing that you have is you have the foundations where you walk through and you basically are looking at the AX and the BX differences on the x-axis. And what foundations has you do, it has you look at 2 equal differences on the x-axis. The only difference between these differences is they are both about 5 oxygen units wide, 5 torr wide on the bottom. But because the curve curves differently, they end up being different y distances. So like the a to the x corresponds to a similarly sized a to the y on the y-axis, but the b to the x corresponds to a bigger difference on the y-axis. That means the same difference in oxygen pressure will result in a greater difference in the oxygen binding, which is the y-axis, right? If the Bx is located on a steeper part of the curve. The main thing here is the steepness of the curve accelerates binding when you are in the steeper part of the curve, which is here roughly between 20 torr and maybe 60 torr or something like that. And that changes how the whole thing works. So that's what you do. You walk through this. You pair it. The answer to number 7 is they're the same. The answer to number 8 is that BY is bigger than AY. That means that you get more difference in binding from the same difference in pressure depending on where that difference lies on the curve. And so this is how you use it in number 9. And this goes exactly to my point. This is the last point I made in class, that try as you might to draw a simple hyperbola, you cannot draw a simple hyperbola that will have a greater difference between the red region in the lungs and the blue region in the tissues. You can't do it mathematically. The way to get the biggest difference between lungs and tissues is to draw a curve that steepens in the middle and that have at least part of the curve lie on the steep part. The PO2 in the tissues is on the steep part, allowing there to be a greater difference between lungs and tissues. And this works out in a certain way on the graph. And there's some things, and they look a little bit complicated when you come across it. But that's what I want to walk you through. And I want to do it this way so that you have the time to walk through it at your own pace. So I'm using the term sigmoid. That means S-shaped. And that means cooperative binding or folding, as we've seen for protein folding. The other thing is that hemoglobin is an allosteric protein. That means that it can adopt a different shape. As soon as you see the difference between the T state and the R state, you can know that that is associated with an allosteric, an other structured protein, other shaped. Hemoglobin is also homotropic. That means oxygen binds and at the same time causes the shape change. Its function is to bind oxygen. Oxygen is also what causes the shape change. These two things can go together, but they don't have to. And you have other cases where you have heterotropic modulators. Heterotropic ones will bind at a different site. That its function is not to bind these, but the heterotropic ones bind and they change its function at another site for a different molecule. Hetero is different molecule. And there are some molecules we're going to talk about that are heterotropic modulators. They are not oxygen, but they change how hemoglobin is shaped, and therefore they change how oxygen is bound. Okay? So for example, actually Oh, here's an example of a different molecule that can bind in the site, can result in the shape change, but it binds in the binding site and it causes a difference in its oxygen binding. So, I have a sentence that is wrong at the top, and I want you to think about what's wrong. Carbon monoxide blocks binding sites and reduces hemoglobin's affinity for oxygen. That's almost right, but there's something that's wrong with it. So I want you to stop and look at that. It's very definitely true the carbon monoxide blocks binding sites. We've already said that. But what's wrong with reduces the hemoglobin's affinity for oxygen? For the statement to be true, it needs to be true at all oxygen pressures. Reducing hemoglobin's affinity for oxygen means that the line with carbon monoxide should be below the equivalent line for hemoglobin at all pressures. Look at that line. The 50% CO line actually is higher in affinity when you're at very low concentrations of oxygen. That's kind of weird, but if you think about it, it actually makes sense with what we know about the molecule. When you get like 100, or in this case 50% of the sites are occupied by CO, and that's irreversible, right? It's never going to come off. So basically they're lost. But they're bound to something that has two things in it, right? Two atoms in it. That will cause the shape change exactly as much as the O2 will cause a shape change. So that means in the case where you have, like maybe, in the case where you have 50% of the sites bound, it's already shifted the other two open sites more toward the oxygen binding level of the R state. Okay, it's shifted it from the T state to the R state because two of the sites are occupied. So the two open sites are actually made more receptive, and that's reflected by the fact on the left side of the 50% curve you actually have a higher Y value. Now it doesn't really matter because that person can still only use 50% of their hemoglobin binding sites at all, and once they are occupied you get a very low difference between tissues and lungs, like about a 0.05 difference in Y between the pink level and the blue level for the 50% line. So the other curve that's shown here, normal hemoglobin, 50% occupied with CO. Then there's the anemic individual, which simply has less concentration of hemoglobin. Okay, that means that their red blood cells are messed up, they have fewer red blood cells, and therefore a lower concentration of hemoglobin in their blood. And so let's say if they have 50% of the red blood cells, then they have 50% of the hemoglobin in the red blood cells, but otherwise the hemoglobin is completely normal. So you see for the anemic individual, you truly have a reduced population affinity for oxygen, in the sense of that sample of blood only holds 50% of the oxygen at all pressures, because there's no allosteric difference. There's no other molecule like there is when there's CO. So look at the difference. You have the same number of available binding sites in the 50% CO sample and the anemic sample, which has 50% of the concentration. But you have different curves. And that makes that statement at the top is maybe 75% right. It's true at all PO2s that are above 3, but it is not 100% true. So watch out for stuff like that on the test. So the one thing that we can say is, how can we draw that cooperative binding line with a simple equation? Well, we quickly find out that it's pretty easy to get started, but it's not easy to finish the equation. So let me show you how to get started. What you do is exactly what we did with myoglobin. We say we have a P plus L reaction going to PL, but instead of just one L, we have 4 L's. So we can rewrite that as P plus NL goes to PLN sub N. And with the association constant for that equilibrium, products over reactants, and the one rule is that our ligand concentration is now not just simply the concentration, but it's raised to the power of the stoichiometric coefficient. That means that instead of PL bracket over P bracket L bracket, we now have a little superscript N. Remember, this is the general chemistry rule of how these equilibrium equations work. So that makes our equation go from this for this hyperbola. The L to the n gets moved around just by algebra, and it ends up being in the hyperbola equation. But you look at that, L is our x-axis. That is no longer a hyperbola. That's a sigmoid. And that gets to some pretty complicated math to deal with that thing. So we can't deal with that equation directly. I can't ask you to take that equation and plot it out and get a sigmoidal curve. If you knew how to do the math, you actually could do that. But it's tricky because you have the n to the power there. But what I can ask you to do is I can ask you to plot a straight line. So if we can turn that mess that's shown on the top here to a simple line on the bottom, then we can actually get I can actually ask you to interpret that. And so what we need to do is we need to use the function of logs, where the log of x to the y equals y times log x. This special function of logs allows us to move the exponent out of the variable and to move it in front of the log. And honestly, I'm not going to ask you to do this yourself, so if you don't remember this, just follow along and trust me on this, okay? But hopefully you remember this and you can see that all this follows the math rules. So we take the mess on the top. What you do, the first thing you do is you just move it over to get rid of the fraction and cross multiply the theta through. And then you end up with the equation that is at level number 2. Level number 2 equation, well, I want to get all my L's on the same side. So I'm going to get my L's on the same side, and then I'm going to uncross multiply. I'm going to factor that out. So now I have all my L's together on the right. That's where I want my x's to be. And that's great, except that I need to get rid of the superscript. I need to get rid of that power thing to get a straight line, which will be by y equals mx plus b. It needs to be x without an exponent, right? So what we do is we move all the thetas to the left, we move everything else to the right, and at the very bottom we take the log of both sides. Once we take the log, what we can do is we can move that n out of the way. We can actually move the kd out of the way because the kd is actually the log of kd to the minus 1. Okay, but the main thing is logs allow us to turn the exponents into a straight line. And once we do that, we just move stuff around. We have this weird log term on the left, but we have all of our theta on the left, which I want to be my y, all my L on the right, and it is without an exponent, which means if I just take the log of it, I get a straight line. And I get a straight line where the slope is n. Okay? That's the one thing I want to show you that. You see that on the bottom? The slope, this is now y equals mx plus b. Y is log of theta over 1 minus theta. The slope is n. Log of L is the x. And minus log of the kd is the intercept. So that means I can do a plot of that weird thing. Log of theta over 1 minus theta. I can do it on the y-axis, and I do a log of the ligand concentration on the x-axis. And just remember, for globins binding oxygen, you're going to need to have that as you can express it as a PO2 as well as expressing it as a molarity. Either one works. And so what this ends up doing is it ends up giving you the slope of the L to the n. The slope of it is equal to the number of ligands that you have cooperating with each other. This is actually a measure of cooperativity. And so what's really cool is that myoglobin, it takes the hyperbola for myoglobin, it straightens that out into the blue line. The blue line has exactly a slope of 1 on this graph. That makes sense. Myoglobin only binds one thing. It only has an exponent of 1. And it ends up working with this because it's a simpler form of the hemoglobin situation. For hemoglobin, we can actually see that there's actually 3 areas for it. Hemoglobin, if we are below the steep section of the curve, to the left of it, we will get the low affinity state that we showed on the previous graph with an n equals 1. If we are at like really high oxygen concentrations on the right side of the graph, we'll also get n equals 1, but with a higher intercept. And remember that the higher intercept corresponds with tighter binding, because the intercept contains Kd. The really interesting part is what it does in the middle. In the middle, for the steep part of the graph, where you're switching from the low affinity state to the high affinity state, the slope for this graph gets steeper too. This is all the same kind of graph. So when the one graph gets steeper, the log graph gets steeper. It just gets steeper by a straight line. And it gets steeper by a line with a slope, in this case, of 3. What's really cool about this is you can take a mutated form of hemoglobin, and you'll actually get numbers that are slightly different than 3. You can take a form of hemoglobin where it is not as cooperative. It's sort of stiffer. It doesn't respond as much to the difference between the T state and the R state. And that hemoglobin will have a lower slope, maybe a slope of 2.8, 2.5 on this graph. That means that the binding sites are not cooperating. When one changes, the others still change, but they change a little bit less. And so you can have a degree of cooperativity. This is called the Hill plot. It is the most complicated math that we'll have. Remember how to make it in the sense of how to draw it and what it means. Remember what N means. You don't have to derive it. I just showed you that so I could show you that it's algebra, but then you have to be able to say what it means. It means that we can measure the cooperativity of different molecules of hemoglobin, and we can get a number that makes sense. There are 4 binding sites. It makes sense that they're cooperating, like 3 of them are perfectly cooperating, or they're cooperating as if there were 3 of them. We don't want to go too much, but it's really useful for comparing different hemoglobins that might have lower cooperativities than 3. So that's what it does. Hill plots turns a sigmoid into straight lines. The slope is the important thing, and it's the degree of cooperativity. It's sort of the number of apparent binding sites. The theoretical maximum is for hemoglobin, I believe, is 4, but the practical maximum is around 3, because you are not going to get perfectly responsive proteins that will completely cooperate. As soon as you add 1 oxygen, all of the 3 other sites open up completely. That doesn't make sense. What you see in real life is you see numbers between 2 and 3. So that means that you can actually do all sorts of experiments. And one of the first experiments that they did is that they ran the hemoglobin binding curves at different pHs, and they saw a significant effect. When you have a lower pH, which means more protons around, you have actually less oxygen binding. A lower pH will interfere with oxygen binding. You can see that the binding is going down as the pH goes down in this case. So why would that be? Actually, you already have a clue for one explanation for why that would be. If you have lower binding, that means you have somehow stabilized. One way to get lower binding is to stabilize the T state. So if the T state depends on a proton being there, if you add more protons, you'll have more T state, and you'll have less oxygen binding because the T state is lower in affinity, right? And look at what we showed you earlier. We showed you the zoomed in thing of the T state. Remember that histidine. The histidine has a pKa in solution by itself. It's around pKa of 6, but it can be shifted. And it can be shifted so that it can be significantly protonated or deprotonated at physiological pH. And if you look at the histidine needs the proton to be able to form the T state. That means if you have more protons, you're going to have more plus-charged histidines. If you have more plus-charged histidines, you have more histidine-asparagine interactions, more bonds, therefore more T-state hemoglobin. More T-state hemoglobin means less oxygen binding. So if a proton goes on at the C-terminus here, then actually it will cause oxygens to come off at the binding site. The more protonated that histidine is, the better it interacts with asparagine FG1, and hemoglobin shifts toward the T state. Now, this is not the only protonation that actually strengthens the T state. They found out that there's like half a dozen of them that significantly strengthen the T state. But they all go in that direction, and they all end up with this, what's called the Bohr effect, because it was found by the scientist whose last name was Bohr. You might have heard of a scientist's last name Bohr, Niels Bohr, in your physics class. This is actually his father. So his father was a biochemist, and he found out that if you increase the pH, you decrease binding. Only later did we really figure out why that happens. So look at that proton. That proton is literally forming a bridge between the histidine and the aspartate. We know that the histidine pKa is going to be close, plus or minus 1 unit or 2, to physiological pH, so it makes sense that it could be something where you add the proton and it forms the bridge, strengthens the T state, weakens oxygen binding. But there's even more. The N-terminus actually has a role to play in binding. It turns out that the N-terminus can bind carbon dioxide, CO2, and this is something you couldn't predict without doing the experiment. But CO2 will bind to the amino terminal residue, and that actually kicks off a proton. So think about what the chain of events is here. More CO2 means you're in a tissue where you have the cells working really hard and putting out the CO2 that you need to get out of there, and they need more oxygen. If they're putting out more CO2, they need more O2. So what happens? The CO2 binds the N-terminus, protons come off locally, protons go back on to the histidine, they form the T state, that weakens oxygen binding, and the oxygen pops off. So you have CO2 coming on one end of the molecule. You have a pH-mediated weakening of oxygen binding, which means oxygen is going to be released from hemoglobin. If you can follow this chain of events, you understand how hemoglobin works. And so, in a sense, carbon dioxide and oxygen have opposite effects. They sort of compete for hemoglobin binding, even though they're binding at different places. Because when one goes on, the binding for the other one gets weakened. And so that's what I've spelled this out. I said it in words on the last slide, so you can just pause this and read this if you want it visually. But just realize we're dealing with these things. When I give you something, you take that condition and you follow it to where it goes. If I say CO2 has increased, then you say oxygen binding has weakened. Oxygen affinity has weakened, and the Kd has gone up for oxygen binding. But if that's true, if I give you another problem that says the opposite, then you're pushing on it from the other direction. So if I say there's a lot of oxygen around, what happens then? What happens then is any CO2 bound to the terminus is actually kicked off by the oxygen. The high amounts of oxygen push on the other direction. They result in the CO2 coming off. So realize with these equilibrium problems, I give you the cause, and you follow it to the effect. If you have higher oxygen, then CO2 gets pushed off, the affinity for CO2 gets weakened. If I have more CO2, the affinity for oxygen gets weakened. So pay close attention to that. That's what makes students stumble a lot in this particular unit. Bottom line, it's an amazing molecule. It's not just a simple hyperbola. It's complex, which means the problems are going to be harder. But this is it. This is what the problems are going to be based on. And so I want to give you this, and I want you to be able to trace the chain. I want you to be able to describe this as a case of allosteric binding cooperativity. And I want you to explain how it works to make hemoglobin be an amazing molecule that is really well suited for the job of transporting oxygen from the lungs to the tissues, and also for transporting CO2 from the tissues to the lungs, weirdly enough. So I have a couple of animated videos for you. I'm going to try to show these to you. If they don't look that good, go to Canvas and we have them there. But I want to show you, there's a scientist whose lab work, she runs a lab basically, but what she does is she runs a dry lab, an in silico lab. Well, what she does is she makes visualizations. She makes good pictures, animated pictures. It's like being a bit of an artist as well. So Janet Iwasa is the name of the scientist, and I'm going to show you the 3 movies. Then you'll be ready to do the homework. So if this works, I will show you the 3 movies. If you go to Canvas, you can see that I have the movies right here. And let's see if this works. This is the first movie. It shows oxygen going on and off. Red is oxygen bound. Blue is oxygen unbound. So that means red is the R state, blue is the T state. Okay. And if you zoom out, you can see that going from red to blue, the whole molecule shifts around. This gives you a better view of what's going on at the C-terminus, by the way. You can see more of that motion in the middle. And oxygen bound, oxygen unbound, and then if you do just the surface of the molecule, you can really see that C-terminus move. It's moving like this when you go from the red R state where it's folded into the T state where it's sort of brought up to the top. So, and now she does it without the colors, so you can really see, this gives you the best view of the motion. And it's formed between the beta subunits. Remember that the interface that changes is the weak interface. So here's another view. Let's take a look at this. Here's oxygen. This shows you cooperativity. Oxygen is trying to bind and falling off because it's weaker. Once one of them sticks, the other shift into the red conformation, and that's an example of cooperativity. Same thing happens in the other direction. You see that if one falls off, the other three are still formed, and so actually the binding stays tight until two fall off, and then the binding is cooperatively weakened. Cooperativity works both directions. Finally, here's one more thing that I want to show you, and this is what the next video is about. There's a video, here's a molecule, and if you look at this, it's between the two beta subunits. So it's located at exactly the point where the C-termini come together. If this molecule is bound there, then the C-termini can't come together as easily. They have to push against that molecule to be able to come together. And so that molecule, you might expect, would affect the way that oxygen binds. And that has medical purpose, and that's why I'm going to use it in the next lecture. That molecule here is called 2,3-DPG. We call it BPG. We call it bi instead of diphosphoglycerate. But more on that in the next video. Make sure that you do not miss it.